My next guest is neurobiologist Professor Jonathan Stone, a leading light in biological science in Australia. He served as secretary in the Biological Sciences Division in the Australian Academy of Sciences for several years and is recipient of numerous science awards. In his research papers, over 200 in all, he asks interesting and important questions. In this conversation, we are going to dig deeply into the ways the brain, our neural network, protects itself from damage, and in so doing, understand how to protect our brains from disease. This biological detective work will also reveal the kind of diet and food components that protect us and why. So, Professor Jonathan Stone, welcome to my podcast.
Thank you and good morning.
Now, I'd like to start at the end and work backwards, so to speak. Firstly, what I love about your papers, and I had no idea that you'd written so many fantastic ones, are the questions you ask. And in the last publication, in the most prestigious journal of Alzheimer's disease, with a provocative title, What is Frustrating the Search for Treatment of Dementia? Fraud or Ideas? You ask an obvious question, and that is why are we evolutionarily designed to produce amyloid? This is a peptide, like a protein, a molecule that is toxic to us. Why do we produce this? It's found in a lot of people with dementia, not everybody, but why do we do this?
So, the question I asked in that short commentary was not why do we produce it if, if it's toxic, which seems to accept that it's toxic, but why would we produce late in life or any time in life a molecule that's toxic to the brain? And that question comes out of classical Darwinian evolutionary theory, which says that characteristics get developed and enter the genome if they help the species. Why would any species develop a toxic molecule sufficient to kill its own brain and put old people through the tragedy of dementia? And the answer turns out to be simple. It's not toxic. Amyloid beta has been described as toxic, reported as toxic, assumed to be toxic. But when you go into the literature, there's a long line of papers saying that actually at low concentrations, it's trophic to neurons, it helps them grow, it helps them form synapses. It detoxifies hemoglobin, which is the oxygen carrying molecule, which has to be kept out of tissues because it's so toxic. But if it gets into the brain, beta amyloid binds tightly to it and detoxifies it. And some people have argued, and I think the arguments are going to prove to be a good one, that it also is they call it a bioflocculant, that it scoops up pathogens that, if there's bleeding into the brain, can get into the brain and cause an encephalopathy, which makes the whole process of brain aging worse. So and my point in that little article was this, that I was responding to a book by Charles Pillar, a distinguished scientific journalist in America, who had argued well, and told the story well, that the search for the cure to dementia was so frustrating that people were committing fraud, and they certainly were. And I wondered whether and he argued reasonably that perhaps the whole field was being slowed down by fraud, and no doubt it is. But my view was maybe it's being slowed down more by the failure of people, including me, to ask questions that any biologist should ask, like the one you started with. Why would the brain ever produce a molecule that killed itself in a slow and brutal way?
So, are you saying, just going back to the hemoglobin? So, we're
Yeah.
talking effectively about bleeding in the brain. And the concept here is that amyloid beta protects us from the dangers of hemoglobin, which, of course, is also very rich in iron and oxygen species. So, is bleeding in the brain common? I mean, of course, we know about strokes where there's a catastrophic bleeding. But is strokes sort of like the tip of the iceberg? Is there a lot of bleeding that occurs in older people into the brain?
Yes, yes, it's a nice way of putting it. If a largish vessel bleeds, then a largish bit of brain is deprived of its oxygen and glucose and will stop functioning, and you'll see it as a stroke. But two groups of people, pathologists with markers and microscopes, and the brain scanners with their high resolution MRI have picked up what are called silent microbleeds. Bleedings from small vessels that the patient doesn't notice and therefore doesn't go and see his physician about it. And they destroy much smaller patches of tissue that are, as the name implies, silent, clinically silent. And the Alzheimer brain is got these in tens of thousands. And at each one of them, abnormal proteins appear, one of which is beta amyloid. So yes, not you and me, Patrick, all the other gentlemen out there of similar age have got some of these small bleeds. And a critical question is, how much damage have they done? And so to touch on the issue you raised earlier of brain protection, what can you do to stop them? And that has kept me busy for the last several years with ideas that I should have realized much earlier needed addressing, but I think I'm getting to address a lot of them.
And before we get into them, so here is the idea that the amyloid beta somehow protects us from these bleeds. Now, in a very linear way, I would ask, how does it do that? Is it like a plaster? Does it actually stop the bleed? What in outline is the amyloid doing that protects us from these micro silent bleeds?
Okay, so when a bleed occurs, 3 or 4 um, causes of toxicity, death to neurons, are set in. One of them is hemoglobin. Hemoglobin should not be in the brain. And we have evolved a barrier system in the vessels of the brain to keep hemoglobin out, just keep it out. Not only that, the hemoglobin is wrapped up in red cells, and so if it's to get out, a whole cell has to get out. Now, usually that doesn't happen. Long decades that doesn't happen, but occasionally, and the evidence is clear, a capillary will bleed. And a tiny amount of blood, less than 1/10 of a millimeter of bleeding will appear. It's toxic. What's toxic? It turns out it's the heme, the iron-containing moiety of hemoglobin, which is heme plus a globin protein. And what's poisonous in there, what's toxic, what will destroy a nerve cell, what will destroy membranes, is the iron in the hemoglobin. And so I got to ask myself, why is it that in mammals like us that are absolutely dependent on oxygen, okay, we've got lungs, oxygen gets into the blood from the lungs and is carried by the blood to the tissue. Terrific. But why is it that the oxygen carrying molecule is so toxic? It's toxic to the brain. And you see it not just in dementia, but you see it in thalassemia and sickle cell anemia, where the red cells break down in the blood, which they also should not do, and causes great morbidity for the sufferers. And that takes you into the chemistry of oxygen and iron. So we're way back now in the evolution of animals. The first animals did not use oxygen, and they got their energy, and they were single cell animals. They got their energy from carbohydrates, and a lot of the energy came from sunlight, which they used down to break down carbohydrates. They did not use oxygen. They were known as anaerobic. But it turns out that those anaerobic mechanisms produced oxygen, and there was 500 million years ago, I think it was, the great oxygen event, when an event, it took 1,000,000 years, but the atmosphere of the Earth accumulated oxygen secreted by anaerobic mechanisms. And then another evolutionary process began in which animals used oxygen in what biochemists call the oxidation phosphorylation pathways. They used oxygen to get more energy, about 10 times more energy, out of a feed of glucose. And suddenly animals emerged, the vertebrates and certainly the mammals, which absolutely rejoiced with that oxygen, and we produced animals like us, and well, you know, the variety of vertebrates from dinosaurs to mice that's been produced. And so they developed all these vertebrates that were absolutely dependent on oxygen. And somehow, if this was to succeed, we needed a molecule in the blood that would pick up oxygen by diffusion in the lungs and release it in the tissues. And it works. It works every time. But it turned out that the chemistry of oxygen is such that you need a powerful metal like iron, IRON, ions to grasp the oxygen, and using molecular forces that are toxic if they released into tissue like the brain. So the toxicity of hemoglobin arose from the fundamentals of our metabolism, and it's still a problem for us today. And so what's happening as the brain ages and these bleeds occur is that more and more small patches of brain are being killed off by this molecule that does the absolutely critical thing of getting oxygen into tissues, but late in life causes this terrible tragedy that we know as dementia.
And the amyloid contains that somehow, the amyloid beta?
Yeah, so amyloid is one of 3 or 4 molecules that have evolved to be expressed wherever a bleed occurs, and not just in the brain, but particularly important in the brain. And it binds tightly to the heme and reduces its toxicity, if I remember rightly, by a factor of 10. And so it's a built-in, okay, there's not going to be a bleed, but if there's a bleed, we've got a mechanism that has evolved and is built into the genome, and it produces the amyloid wherever there's a bleed. And that's why several insightful biologists, American biologists, commented 25 years ago that the reason amyloid is so present in the aging brain, and particularly so in the dementing brain, is that it's not it. They put it this way: said it's not it's not the arsonist. Amyloid is the fireman.
So it's not the fire, it is the fireman.
Yeah.
Such an important concept. And before digging a little bit more into that, one of the hot areas of another group of toxins that enters the brain are microbes. We've been hearing more and more about gum disease and P. gingivalis, and recently certain forms of herpes from the gut that get into the brain. Is this another sort of situation where amyloid is triggered to protect against pathogenic microbes?
Yes. Well, as I mentioned earlier, the people who've investigated it, so it's a small literature, but it's got it'll last. It'll stand the test. These people argue that one of the roles that amyloid plays is to somehow scoop up and wrap up microbes, so bacteria or viruses, at the site of a bleed. Why is that good for the brain? Because if these microbes get into the brain after being kept out of it for the early decades of life by the blood-brain barrier, if they get in, they'll infect the nerve cells, the key cells, and also getting through riding the same bleed will be killer T cells, which will recognize these neurons as infected and kill them. So one of the four mechanisms that come into play and destroy neurons where there's a bleed is immune cytotoxicity. The immune system does what it's meant to do. It finds infected cells and kills them to protect the body from disease. But because the brain cannot effectively resupply late in life the nerve cells that are slowly dying off, that hurries along the process of dementia. And I think that literature will work out. So it's a second positive role for beta amyloid. Yeah.
Now, just to clarify a few terms for those listening, because dementia is diagnosed on the basis of a cognitive function, a decline in cognitive function. Alzheimer's specifically is diagnosed by a brain scan of the central area of the brain, the medial temporal lobe or the hippocampal thickness, and accounts for something like two-thirds of dementia is Alzheimer's if a person has that brain scan. Maybe 17% is called vascular dementia. And our head of research, Dr. Tommy Wood, was explaining that they're very similar. Vascular, could relate to a stroke or a transient ischemic attack. That often the decline in cognitive function is kind of step effect. Suddenly a stroke, a big drop, or a TIA, another drop, and so on. But at the end of the day, you end up in pretty much the same place. And those two already, and mixed dementias, by the way, is often a diagnosis given for somebody with Alzheimer's pathology and vascular pathology. And I think, like you were saying, that the brain scanners find hundreds, if not thousands, of little blips in the brains of people with Alzheimer's and vascular dementia. Is that sort of broadly speaking a good overview of the pathology?
Yeah, and you've made the insightful point that although some dementias begin with memory, and that's commonly called Alzheimer's, some begin with a breakdown of personality. Memory's all right, and they're usually associated with damage in the frontal lobe, the prefrontal cortex. Other neurodegenerations begin like motor neuron disease with very motor symptoms. And there are several other small, rarer forms. There's another one which begins with motor and memory and personality all degenerating at once. It's called multiple system atrophy. So that differential diagnosis, and there were other ones, even in Alzheimer's day, 130 years ago. There was syphilitic dementia and vascular dementia. And it matters what you call things because I know why they called vascular dementia because it occurred after a stroke. But calling it vascular dementia implied that the other dementias weren't vascular, and they've turned out to be, and that's held the field up. Another case where ideas can hold the field up.
And yeah, sorry.
You also made the point that they all end up with an advanced dementia and death. And in my view, although it's still a moot point, I think those several forms of degeneration depend on where the damage starts. Does it start in the frontal cortex? Does it start in the temporal cortex? Does it start in the motor cortex? But it's the same pathology, which is small vessel bleeding, and as it progresses, they all converge on advanced dementia. Does that make sense?
Yeah, I was really blown away by a paper by a man called Teng. He was actually looking at homocysteine. Homocysteine is raised in people who lack B vitamins, especially B12. And we look for odds ratios, it's called. So, for example, if you have twice the risk from, for example, raised homocysteine, that's interesting. In this case, it was a tenfold increased risk. But what really struck me was a 17-fold increased risk in cerebrovascular small blood vessel damage disease in people with raised homocysteine. This is this is really, really high. And that's why spending this time on the vascular side of things makes sense. The other point I want to make for those listening is not everybody who gets diagnosed with Alzheimer's has a lot of amyloid in their brain. And not everybody who has a lot of amyloid in their brain necessarily gets Alzheimer's. So it's not a perfect fit. And I haven't really seen evidence yet to say that it is amyloid beta itself is causative. However, there is this obsession about amyloid such that pretty much the only treatment that is being pursued with significant vigor are injections of antibodies that target amyloid. And in this context, I would say target the fireman, not necessarily the fire, and remove it. Is that a good idea?
No. And if you read the reports that the big pharmas are producing on their drugs, which they hope will get such a boost from a successful outcome, they loyally and honestly report side effects, which include death, brain edema, and cerebral bleeding in, if I remember rightly, a minority of patients, but quite a minority, and 20%, which is about what you'd expect to happen if you pulled amyloid beta, understanding its protective effects, out of the brain. And the positive effects that they report are so limited that the clinician can't detect them in a patient. It's not in a single patient, and the patient and the other carers can't detect it in a patient. And they got significance by getting huge numbers and getting the p-value down below 0.05, but no clinical benefit to patient or carer. Yeah.
Well, what I hadn't realized was the whole sort of amyloid concept emerged from when the gene sequencing showed that some very early onset dementia called Alzheimer's, there was a gene, the amyloid protein precursor, the gene that makes this amyloid protein precursor. So it's called the APP gene. And what I hadn't realized until recently is that there were two clinical studies giving the anti-amyloid injections to the people who do have this very rare, because I think there's 500 families in the world who have this um, amyloid protein producing, mechanism, genetic twist. So they've got high amyloid, and these anti-amyloid drugs, in one of them, failed to produce a significant effect, and in the other one, it made their cognition worse. But once again, a significant minority had bleeding or swelling. So it,
Yeah,
it kind of didn't work in those who were already, producing tons of amyloid for whatever reason. It always
yeah.
struck me that it was unlikely to work with it in a broader group, which is in effect kind of what.
Yes, that was the scientists who did that were pioneering molecular genetics in the 80s and 90s, and they were so far ahead of the field in understanding that somehow beta amyloid was involved. They uh, I remember going to a seminar where the speaker said, look, in the families we've investigated with this early onset dementia, where everybody gets dementia early, it's highly penetrant mutants. We've identified 3 of these mutants, and they all have something to do with APP, the amyloid processing protein. And he's absolutely right. He was absolutely right. And I remember a discussion breaking out, and somebody in the audience saying, he said, to me, this absolutely proves that amyloid beta is the toxin. It must be. And of course, he missed the fireman possibility. So I was influenced by the similar papers, and I remember nobody else seemed to comment on it, but what they do is they show the family trees, and they show that generation after generation breaks down and dements. But I noticed in one family tree that a quarter of the descendants who were suffering, they suffered not dementia, but what was clinically called stroke. And it seemed to me even then that this could be a blood vessel weak fragility problem, and that, yeah, sometimes small vessels bled, sometimes large vessels bled. And I don't know why my mind grabbed on that, but it was a couple of decades ago, and it said to me, actually, this could be a bleeding problem. And every time I pick it up, have a good look at it from a new point of view, yeah, dementia is a bleeding problem. Yeah, it's a vascular dementia of small vessels. Mm.
Now, a slight tangent on this, but I want to tell you that I was the last person to film Dr. Linus Pauling before he
Oh, yeah.
died. In his 90s, he was one of many scientists that I've had the great pleasure of working with, um, some still alive, some not, in their 90s, minds as sharp as a razor. And he got obsessed by vitamin C. And there's one thing biologically that I've never really found the answer to. Vitamin C is made from glucose. So it's a relatively simple, I think it's a 4-step process that results in ascorbic acid. So it kind of, and with this iron, I'm thinking oxidation. The iron is dangerous in the brain. It's a
Yeah.
reaction with oxygen and iron oxidation. And here we have a biology. There's oxygen-based life forms need glucose plus oxygen to produce energy, and produce these, toxic oxidants. However, with every bit of glucose that we make, I mean, us humans, we have a bit of a problem. We've lost the ability to synthesize vitamin C, but almost every other animal can do it. Also, there is this production of an antioxidant. vitamin C. It got me thinking of the idea that in order to have an oxygen-based life form running on glucose, generating lots of energy, there must have been built into that mix the exhaust mechanism that is ascorbic acid, helping to protect against the adverse exhaust fumes of the process, so to speak.
But,
Do you think that's true?
um, it's a bit of the biochemistry that I've never quite mastered, but the the oxidants are so damaging, that there has to, you absolutely swear that there would have been an evolutionary reaction of antioxidants, and they've been well documented. Yes, I think it's true.
And before we move on to your second really important point about resilience, people are
Mm.
going to be asking, how do I stop having any bleeds in my brain? What is it that protects my vasculature? Obviously, as a nutritionist, well, Hippocrates effectively said what's good for the heart is good for the brain. So, when we hear about, wonderful antioxidant-rich foods and eating a rainbow diet and omega-3 seems to be very important in blood vessels and vitamin E and vitamin C and all that sort of thing. But yeah, in a nutshell, for those who want to have really good vascular health, what should they be doing? Obviously, not hitting their head a lot, boxing, or having lots of headers in football.
Head traumas, head traumas, toxic, and and yeah. So the game, the football form that's known worldwide as football, but it is soccer. It's one of the least impactful of the footballs, and yet players suffer the dementia of trauma, and it's almost certainly from heading the ball. Probably not from heading the ball in matches, but they must train, and So look, we already know, all of us, that exercise, people who exercised dement later, people who eat vegetable-rich diets dement later. There's quite a strong literature that says that if you experience intermittent hunger, which in the wild is natural, but in modern humans, it's something like the 5-2 diet. You dement later. There are certain radiations, infrared in particular, which seem to produce a delaying effect on cerebral bleeding. And I think the literature is very strong that all of these do something to the tissue that makes it more resilient and the vessels more resilient. It's not an all or nothing thing, but it's measurable. So let's consider the dietary one. The data are unassailable that eating a Mediterranean diet, or whatever you want to call it, rich in vegetables, delays the onset of dementia. And when my group looked at it closely, we asked what it is about vegetables that's not there in, or I suppose you call it the Western diet, which is rich in meat and so on. And it turns out that all plants in the competition for a bit of sun and a bit of soil produce toxins that are designed to make their predators, caterpillars or birds, or their competitor plants sick. So if you look under some trees, like fir trees in a northern forest, or under the Moreton Bay figs in Sydney, very little grows. And this is because the trees are producing toxins that say, this is my bit of soil and I'm going to kill you if I possibly can. And they're there in all plants. So we've evolved to eat certain vegetables. They too have toxins that they produce to inhibit everything around them that's alive. And
In
they
my
have also,
garden, in my garden here in Wales, I have a Pendragon apple tree, which has very high salvestrols, resveratrol. It's the same sort of thing that
yeah,
these are produced. They're sort of to protect the plant from attack,
yeah,
such as
yeah,
fungi. And I love this concept that you introduced in your paper of trace toxins, because many of the things that we wax lyrical about, curcumin and turmeric, for example, maybe quercetin, resveratrol, all of these sort of new fancy things which don't fit into the category of a nutrient. It's not a vitamin or a mineral
oh,
or
I
something
know.
that is
Oh,
essential. And yet they seem to be very protective. And what you're saying is that these toxins produced specifically by plants to protect them also seem to protect us. How,
yes,
how do they do
yes.
that?
And we didn't know about it. We didn't know about it till I don't know, this maybe this century. There wasn't much literature on it in the 20th century. And it's such a sort of silent system. It's there, it works. You can see it in the epidemiology. Anyway, long story short, one way you can slow the bleeding down is to make sure there are plenty of um, vegetables in your diet, or you can go and buy them in these days in the health food store. Take 3 capsules in the morning, eat what you like for the rest of the day. Not in unwanted quantities, but there's nothing wrong with beet. It seems to be free of any toxins. Animals don't seem to have done what plants do and produce toxins to discourage other things from eating them. But yeah, so one thing you can do is make sure you get this component of your diet, which we call trace toxins. And meal after meal after meal, it really works. Exercise. Now, I once asked my cardiologist, why is it the high blood pressure is bad for you, but exercise, which puts blood pressure up, is good for you? And I didn't know, she didn't know, but that was decades ago. It now emerging that exercising muscle, particularly if it's hypoxic, produces myokines, they're called, a whole range of growth factors that are released into the bloodstream, circulate to all tissues, and increase the resilience of tissues. What's the resilience of tissues? It's the strength of our protein membranes to resist breakdown forces. And there's another one I've better mention, which is heat. Back in the 20th century, biochemists demonstrated a form of protein, they called them heat shock proteins, that are expressed if our temperature in mammals goes up a degree or two, as it does in saunas. And these turn out to be chaperones that bind on to other proteins and stabilize them. So people who have that Scandinavian habit of soreness also live longer without dementia. So I think that's 5. The toxins of plants, exercise, intermittent hunger, light rays, and heat. Yeah. So there's quite a few things you can do, and the only one I hate is hunger. I don't like it.
But it's a bit like we're talking here about the whole concept of resilience. If you like, putting money into your health deposit account. And you do raise that very important question in your recent paper, which is why are treatments relatively ineffective in those who've already got dementia? However, we can prevent things. I'd like to return to that in a minute. But it sounds like if it doesn't kill you, it makes you stronger. It's a little bit like that. That
Yeah,
exercise is a
yeah.
stressor, but there's a reaction that occurs in the body that makes it stronger. Not eating, hunger is a stressor, but it seems to, do us a lot of good. And these toxic compounds in plants, many of which are toxic in too much quantity. So
Oh,
there's
yeah.
a quantity effect, but actually it stimulates some response in the body that puts more money in your health deposit account, gives you more resilience, and protects you from dementia. Is that kind of how you see it?
Yes, yes. That's nicely put.
So, yeah, resilience. So, here is this other point that you make. And it's kind of an irony because I remember being with Professor David Smith, who was the Vice Dean of Oxford Medical School, at the point where he was breaking the code in his double blind randomized trial on B vitamins versus placebo. And yeah, it was. A very exciting moment when he found that those who had the higher amounts of B vitamins, or that given the B vitamin supplements with raised homocysteine, had a massive reduction in cognitive decline, and also in the rate of brain shrinkage. And yes, so the study was criticized because the people in the study had pre-dementia, mild cognitive impairment. And the critics said, we don't know it's going to help people with Alzheimer's because you're starting too early. Anyway, a few years later, all the trials failed pretty much in people with diagnosed Alzheimer's. So then pharma sort of backtracked and said, we need to treat people much earlier. And that's the point you make. Why are these things failing late in a disease process when we can pick up so many protective factors and protect people earlier? And that's the whole critical point of resilience. Do you want to elaborate on that?
Yes. So the the question that we had to ask then to get at that was why does why does dementia occur so late? What's stopping it from appearing? Um, Even though we know that the what other people thought was the key pathology, these little senile plaques with beta amyloid in them, can occur much earlier and can occur in people. Yeah. So others developed the idea in various ways. The way we developed it, and I'm meaning to acknowledge those who thought before us, is some of them said, look, if this key pathology is appearing, but there's no dementia, there must be a reserve. Sometimes they called it cognitive reserve. I'm an anatomist. I called it brain reserve, brain tissue reserve. Um, and I was very influenced because there was one of our studies in which we wondered whether capillary bleeding was also causing idiopathic lung pulmonary fibrosis and chronic kidney disease, which tend to occur with dementia late in life. We thought it did. And for the kidney, for example, we're born with two nice fat kidneys. But we also know, because occasionally people are born with one or lose one in trauma, that the young human doesn't need two kidneys. He does fine on one for decades. And there must be that we get two nice rounded kidneys. They do fibrose later in life. We must have a reserve of kidney tissue. That as the kidney slowly fibroses throughout life, as it does in everyone, gets brought into play just to keep kidney function going. So, and there is a concept in the literature of renal reserve. And there's a similar concept for lung. You can do without one lung if you have to and live for decades. And I thought, is there a brain reserve? There must be. Others have thought it. And then, of course, we had to deal with the question, where is it? Because it's not in the two lobes of the brain. They're not two hemispheres of the brain, and they're not equivalent to the two kidneys. They have specialties. And we argued that it's there in the thickness of the cerebral cortex, where all the literature says that cognition resides. So, question, does the cortex get thinner with age? The answer is it does. And is that true in everyone? The answer is yes. No one escapes that. Cognition can escape it. All right, is there a literature which says that cognition is preserved in people in which the cortex remains thick? And the answer is yes. It wasn't expressed like that. People who were still cognitively going in their 90s and as centenarians were called super ages, and papers appeared saying super ages have a thicker cerebral cortex. There seems to be a simple correlation between the thickness of your cerebral cortex and whether you're dement or not. Now, there are two implications of that. One you've mentioned several times, which is that's the good cognition bank that you can invest in by lifestyle choices. But also, there must be a limit, because we are seeing people lose it. And that is, they dement, and you can very difficult to get any improvement in those who dement. Not impossible, but very difficult. And my view, my colleagues' view, is that dementia becomes diagnosed when the reserve tissue in the cerebral cortex is exhausted. And because it's exhausted, and the pathology that keeps it, that thinned it in the first place, continues, there's no way back. There's no way back from a diagnosis, or a very slight way back. So the hope for a cure was a bit of a dream. So you must act early, at least from midlife. You must do all you can reasonably fit into your life to protect your brain. And there's a third implication, which is was a bit bleak, which is that everybody will dement.
Yeah, I'm not sure I accept that. I mean, the point is, I remember, I mean, it's a question of what gets you first, because the man who turned me on to Dr. Linus Pauling with his 48 PhDs and 2 Nobel Prizes, who was as sharp as a razor until the end, was a lovely fellow, Dr. Abram Hoffer. And he was head of psychiatric research in Canada. And yes, he after he was 90, it was very hard for him. They didn't really want him to have his psychiatry medical license. So he became a nutritionist and kept doing what he had always been doing. And then 2 weeks before his death, in his mid-90s, he stopped seeing patients. And 4 days before his death, and I was writing to him, we had many conversations. 4 days before his death, he said, I'm not feeling very well. And 2 days before, he checked into the local hospital, and they couldn't actually find any disease as such. But in the last 48 hours, his organs just slowed down, no pain, at peace, no drugs, and packed up. So I stand for the possibility that we may lose our organ reserve and we may lose our hair, but we don't necessarily need to lose all our memories.
Fair enough. Shall I respond to that?
Well, I think one thing we do obviously share is that rather than trying to shut the door once the horse has bolted and wait for a miracle cure, that the focus absolutely needs to be on prevention, on doing the right thing, certainly from midlife. Yes, I'd love to hear your response to that.
So I totally agree on the latter point. you can dement at 70 and you can dement at 100. That's a lot of life to fight for. Yeah. And everybody will make their own decision. There's an old American bit of Bravo of live fast, die young, and leave a good-looking corpse. I'm not as brave as that. So on the inevitability of dementia, my response is that if you take the human population in 5-year groups from 60 to into the 12th decade, the proportion of people with dementia doubles every 5 years. The 90s, you're in the about a quarter of people are demented. The next decade, the centenarians, it's up in the 40%. We don't have data from the 12th decade, but if the doubling every 5 years holds into the 12th decade, then we're pushing near to 100% will be demented. And what happens, of course, is that the brain is showing this great longevity, but some other organ fails. Yeah.
So at our charity, foodforthebrain.org, we have a free online cognitive function test that anyone can do. It's followed by, and please don't say, I don't want to do it because I don't want to know, because it's followed by a questionnaire which works out what your future risk will be if you keep doing the same things. Remember, the definition of insanity is to keep doing the same things and expect different results. And it highlights exactly what to change in order to reduce your risk in the future. So it's very, very worth doing. And I would also like to say that Professor Jonathan Stone, even if you just look up his papers in the last 10 years, they are absolutely fantastic. They ask such good questions. And it reminded me of the quote of the philosopher Wittgenstein, who said, thinking is a gift that is given to one who is worthy of it. The concept is that if you ask the right questions and you keep going, you will get to some significant truths. And Jonathan, I think you've really done that in your career. And I hope that many people read your brilliant papers on trace toxins, on resilience. There's, the latest one, is it fraud or not asking the right questions? That's important. And I think there is one, the brain is destroyed by the pulse. That was a very good paper talking about this whole, vascular issue. So any final words you'd like to give to our listeners? You'll probably get about 3,000 people listening to this podcast.
Well, first, thanks for your generous comments. It's the Greek philosopher, mathematician Pythagoras thought there were 5 stages of natural life, and the 5th of which was senile dementia. And he said it began at the age of 81. And when I read that last year, I thought, yikes, So I've been given a bit of a gift beyond Pythagoras's estimates. It was very like Shakespeare's 7 Ages of Man, the last being mere childishness and oblivion. Yeah. I've been lucky, and I'm lucky to have your generous interview, and thank you for the opportunity.
Thank you for your literally lifetime of very important work. Thank you, Professor Jonathan Stone.
It's been a privilege.